kind of an odd lecture today because we're going to be covering two opposite poles of the subject of neuroscience. One is gross anatomy, namely what has been learnt down the years about the basic structure of the brain, and we'll be talking about really some very obvious structures and some very simple concepts. There'll be plenty of, although you do have to memorize these for the first exam, it's not a lot, and hopefully as the course goes on, you'll become completely happy with where these structures are and what they do. So I'm just going to sort of touch on some basic questions in the first exam related to this material. And then you'll cover all of these parts of the brain sort of in context as we move along. So the nervous system has, incidentally, some people are putting stuff on the discussion pages. There's a nice question about intracellular and extracellular recording, which sort of gets, in my reply, I sort of get into the sort of difference between electrical potential and ion flow and electrical current, and how one sort of refers to them in an answer. So read that one, and also I think there's a couple of others. There's one on glial cells, on astrocytes, which is kind of interesting because I went into the literature and found an actual concrete example of how glial cell regulation of neurotransmitter levels results in epilepsy in mice, where the neurotransmitter uptake proteins, or one of them for one neurotransmitter, is deleted in the genome, it's removed, and the mice are susceptible to epilepsy because they're not lowering the levels of that neurotransmitter. So track through those. None of that is examinable. The concept is, of course, but not the details. But I know you like details, and also like real-life examples, so it's in the discussion. So go there. The subdivisions of the gross anatomy of the nervous system are fairly simple. There's the peripheral nervous system, which includes the sensory neurons that we've looked at already, the axons of the sensory neurons, and their dendrites and everything, and also the motor neurons that we've looked at before. So the peripheral nervous system consists of sensory neurons and motor neurons. And then also the sensory neurons and motor neurons travel along nerves. And in our nervous systems, there are not purely sensory nerves and purely motor nerves. A nerve is a bundle of neuronal axons, and it can contain both motor neuron axons and sensory neuron axons. So the nerves that travel down the length of our arm, from the spinal cord to the tips of our fingers, not only carry information from the fingertips to the nervous system, but also contain the motor neuron axons that command the muscles. And then outside the central nervous system, in the peripheral nervous system, are these objects called ganglia, and we'll meet one in a moment. But ganglia are collections of neuronal cell bodies outside the central nervous system. At least that's the sort of the part of the definition that's true here, completely true, is collections of neuronal cell bodies. They often occur outside the central nervous system, but anatomists have a tendency to also call areas within the central nervous system where neuronal cell bodies are concentrated. They also have a tendency to call those ganglia in some cases as well. And we'll meet an area like that in a few minutes. Okay, so this is sort of not an exclusive definition concerning outside the central nervous system. And then one of the important things to realize about the peripheral nervous system is that while we have one sensory division, only one, a single sensory division, that carries information from both the skin and muscle and joints. While we have just one division of our sensory nervous system, we have two divisions of our motor nervous system. And motor fibers, motor neurons, and motor axons can be categorized in the peripheral nervous system as to whether they innovate, as to whether they target visceral muscles, that is, muscles that control the rumbling of our stomach, muscles that control the diameter of our iris, muscles that control sphincters, for instance. These, and also muscles that, and also the control of glands and hormone release. So all of these aspects of motor coordination are carried out by something called the autonomic or visceral division of the peripheral nervous system, the motor peripheral nervous system. And then the other part of the peripheral nervous system is called the somatic part, and this refers to the motor neurons that drive voluntary muscle movement. So we have sort of a voluntary and an involuntary motor peripheral nervous system. And the cell bodies for these neurons in the autonomic or the somatic peripheral nervous systems tend to be in slightly different places, for instance in the spinal cord, and they're subject to very different commands, obviously. And then, so that's the peripheral nervous system, right? Sensory neurons, nerves, ganglia, and motor neurons, either the autonomic or the somatic divisions. And then there's the central nervous system, which is essentially the brain and spinal cord in our case. So here's a picture of both the central nervous system and the peripheral nervous system. The central nervous system is colored yellow, so you've got your brain, and you've got your spinal cord, and then the peripheral nervous system consists of all the nerves that arise in the spinal cord here and travel out to various parts of the body to drive muscle movement or to pick up sensory information. Okay. And then up here are these so-called, these are the spinal nerves which originate in the spinal cord. There's also cranial nerves which originate from the brainstem, by and large. And these drive the musculature of the face. They convey information from the face and the head to the central nervous system. Okay, so there's no huge difference in function between the cranial nerves and the spinal nerves. They're both sensory and motor, but they just originate in different parts of the central nervous system. So let's look at the thing in more detail here. So here's the brain, and here's the spinal cord. We'll meet all these structures later. Here's the spinal cord, and you see that the spinal cord is divided into segments. All right, and each segment here this is a segment. Each segment has a little sort of protrusion attached to it, and that's called the dorsal root ganglion. And from its name, the dorsal root ganglion here is a collection of neuronal cell bodies. And we've met this guy before, we haven't really talked about it, but this is where it happens, it so happens that this is where those sensory neurons involved in that reflex arc that we looked at, this is where those sensory neurons have their cell bodies. So the dorsal root ganglion is a collection of sensory neuron cell bodies, by and large. Okay. And then from the dorsal root ganglion, sensory nerves will spread out throughout the body, as shown here. So these might be sensory neurons spreading out throughout the body. And then the motor neurons come back, and they enter the spinal cord through the ventral root. Right? For the motor neurons, as you've seen in that reflex, their cell bodies are actually within the spinal cord. So the motor neurons cell bodies are located in the spinal cord, but there's a little pathway for them to leave the spinal cord on the other side, on the ventral surface. Again, we'll see all this later in great detail. So these divisions can be divided into cervical, the neck, thoracic, the chest, the lumbar, the lower abdomen there, and the sacral, right down here, sections. And the nerves that originate from each segment of the spinal cord innervate a very particular part of the body. So these lines here are the divisions between the areas of the body that are innervated by the nerves that originate in these various parts of the spinal cord. So, for instance, all of these areas here are innervated by the cervical neurons in the cervical segment of the spinal cord, both motor and sensory neurons. So it would be motor neurons here that drive movements of the neck, for instance, all right, and the upper chest. Each segment, though, will only pick up sensory information or control the musculature within a certain band of the body. And these bands are referred to as dermatomes. Right? We'll meet them again when we talk about the arrangement of the motor system. You can see immediately that a physician, by looking, for instance, as to whether you have motor control or sensory, are able to feel touch, for instance, within one of these dermatomes, is able to map out where an injury might have occurred within the spinal cord. So, for instance, if you're missing sensation in your feet, it's likely to be an injury down here in the sacral part of the spinal cord. So these dermatome maps are really quite important for physicians and EMTs. Now up here is the trigeminal ganglion. This is the equivalent of the dorsal root ganglion for the spinal nerve, for one of the spinal nerves here, and it innervates the musculature and receives sensory information from the face and the ears. There. Okay. Not the auditory part of the ears, but sort of the surrounds of the ears. So if you can waggle your ears, that's the ganglion that you're sending information through. Yeah.
Does an injury in the thoracic region have an impact on the lumbar and the sacral region?
Yeah, well, it depends what the injury is, but yes, I mean, because there are obviously, for the brain to command movement in your toes, the brain has to send action potentials down axons all the way down the spinal cord. And so if the spinal cord is damaged here or here or here or here, there's a fair chance that everything below that level will be damaged, or the sensation or the control of muscles below that level will be damaged as well. Okay, it may be that the damage is not within these fiber tracts, or is not severe enough to completely remove sensory, delete sensory information, or delete motor control. But obviously you can see that obviously a high cervical section is the worst. All right, and that's the kind of what was his last name was Reed, Christopher Reed. Is it Christopher Reed? Reed? Yeah, Reeves. Yeah. Yeah, he fell off his horse, I think, and suffered a cervical transection here, and lost the ability to sense information or really or motor control down there. So this is sort of the flow of information through the nervous system as a sort of a block diagram. So you have your internal and external environment, the sensory receptors, as we've seen an example of with the stretch receptors in muscles. The sensory receptors send axons through the dorsal root ganglia and into the spinal cord here. So this is the flow of sensory information. And as I mentioned, the sensory information can come from the viscera, it can come from muscles, it can come from joints, and it can come from the skin, obviously. All right, for the spine. And then, so that goes up here. In addition, you have your spinal nerves coming in, and things like the optic nerve or the auditory nerve. And then you have your brain here, which we'll look at in more detail in the next slide. And then the output of the brain travels through the spinal cord, and then the output arrives at either the visceral motor neurons here, which project and innervate and drive smooth muscles, cardiac muscles, and glands, or the information travels down the spinal cord to synapse onto somatic motor neurons, and these then send axons out to the skeletal muscles. So here's this division of the motor system here into two components. All right, so that's the sort of block diagram of how the central nervous system and the peripheral nervous system sort of interrelate. Okay. To do anatomy, you have to get used to directions. And one of the unfortunate things about the human brain is that there's a sort of a kink, a directional kink in the structure of the brain. If we were dogs, right, or cats, or some other nice, large, furry animal, our brain would be essentially in line with our spinal cord, right? But as it is, as humans, since we're bipedal and we look ahead, right, we've evolved, and some of the primates as well, obviously, have evolved a sort of a bend in the central axis of the nervous system, so that our brain is tilted forwards, all right, at this point here. This is the sort of bend, all right? Okay. And so this creates a problem for comparative neuroanatomy when it comes to comparing directions within our brain to directions within a mouse brain or a rabbit brain or any other kind of, or many monkeys' brains. So there are two sets of coordinates, right? The first set of coordinates are rooted in the environment. They show up, which is called superior, right? That's the ceiling, right, in the environment. Inferior is the floor, all right? And posterior is behind us, and anterior is in front of us. All right? So those are a set of coordinates which can be referred to, but which are based on sort of geographical directions, local geography, as it were. All right? Up, down, behind, and in front. And so this is the anterior part of the brain here. Right? This is the posterior part of the brain here. This is the inferior part and the superior part of the brain. Okay. And then there are another set of coordinates, and their usage is kind of overlaid, unfortunately, on people who will refer to either. And that refers to the sort of classical,
Is this something you also find in primates?
Yes, they would have, some of them have the same kink. So it would be, obviously, it's easy to compare a human brain to a chimpanzee's brain than to a mouse brain, for instance. But the other set of coordinates concerns the sort of general layout of vertebrate bodies, and it refers to the dorsal and the ventral side. So the dorsal side of your body is your back, and the ventral side is your tummy. I have a big ventral side. Right? So that's dorsal and ventral, right? Dorsal and ventral, right? And you've probably done dissections of animals and either dissected from the dorsal side or the ventral side, all right? It's sort of a basic dissection sort of descriptions using dissection. Now, because of this kink in the sort of axis of the nervous system, the dorsal side of the brain is not over here, it's actually up here, right? So the dorsal direction with reference to the brain is this direction, quite close to the superior. So you can use sometimes the dorsal brain as being equivalent to the superior part of the brain. Whereas in the spinal cord, dorsal is out in this direction. So it's more or less posterior. Okay, so when you see when an anatomist labels a section of the brain or a part of the brain or a structure in the brain, he may call it, the superior colliculus, which is a set of cell bodies in the brain. It's called the superior colliculus, which means it's probably, it's above the inferior colliculus, right? So that's superior and inferior, right? Or they might refer to the dorsal root ganglion that we've talked about. And that would be a little bunch of ganglion of cells that is located on the dorsal side, just outside the spinal cord. Okay? Or
What about the rostral?
We'll come to those. Right? So the two are used interchangeably, and you just have to be familiar with them both. And thanks for reminding me about rostral and caudal. So rostral refers to the front end of the nervous system, right? And caudal refers to the rear end of the nervous system. So the caudal you can think of as being closer, a caudal direction is closer to your tail, right? Or a tailbone, all right? So it's closer to my tail, all right? When rostral is further towards the front of the nervous system. But again, because of this, so with the spinal cord, it's easy, right? Caudal is this direction, towards your tail. Rostral is up in that direction, up towards your head. But when you come to the brain, things again get shifted. So the rostral now is out in this direction, towards the front of the brain, whereas caudal is out in this direction, towards what would be, in a dog, for instance, the back of the brain, the hind end of the brain. Okay? So, yes?
So in the brain, rostral essentially correlates to anterior? Because aren't they both in the front? And then...
Yeah, rostral correlates with anterior in the brain, but it correlates with superior in the spinal cord. Okay, so you just have to take these, two systems and make them coexist in your mind. So that when somebody refers to a direction in the brain, anterior, posterior, caudal, rostral, you sort of know what they're talking about. Can't do anything more than that. You just have to sort of mentally image it. Right? There are no acronyms. There are no simple solutions. Okay, life is a little bit more, a little bit easier when it comes to sectioning the brain. A horizontal section of the brain is pretty easy to imagine, right? You just it's like slicing the top off an egg. You just take the brain here and you slice in that direction, right? Horizontally, with respect to the floor. All right? That's a horizontal section. So this is superior and inferior and frontal, the front here, the anterior and the posterior back here. All right, so that's a horizontal section. A coronal section is one where it's rather like working a bagel slicer. All right, you put the bagel in and you slice it vertically. So if you put me into a bagel slicer and slice my brain vertically, that would be a coronal section. All right, so the coronal is going to divide my brain into sort of the rostral and caudal sections. And then you have a sagittal section. This is the one that's perhaps most difficult to remember. The sagittal section is one that just comes straight at you through your middle of your eyes. Right? That's a sagittal section. All right, so it's going to split your brain into left half and a right half. It is sagittal section. Okay. And of course, most cases when you make a section of the brain, you don't make one cut, you make two. So you get a thin slice of the brain that is oriented according to these three planes, right? Horizontal plane, the coronal plane, or the sagittal plane. We'll see some examples of this. So before we start sectioning the brain, let's just take a look at it here from the outside. This is where the spinal cord exits down here, and so that the kink in the axis of the brain is buried here inside. And the first thing you see is this huge convoluted area, these two huge convoluted hemispheres, one on each side, one on the left and one on the right, which are known as the cerebrum. Okay, so these are the cerebral hemispheres. So that's this whole big structure here is the cerebral hemisphere. And the cerebral hemisphere is wrinkled. Right? And the wrinkles create valleys and they create peaks on the surface, right? Because they're wrinkles, all right? Like the surface of one of those dogs' heads. Samoyed dogs, those wrinkly dogs with all skin. Looks like that, all right, yeah. Okay, and the peaks here are called gyri, right? The singular is gyrus. And the valleys are called sulci. Singular is sulcus. Right? So you have Precentral sulcus is this valley here, because it's just in front of the midline of the brain. And here's the central sulcus here, just behind the midpoint of the brain here. And here's the post-central gyrus, this sort of peak here, this lump here, and so on and so forth. Okay? And then when a sulcus gets too deep and too large to be labelled as such, when it's very sort of large and special, and actually divides two lobes of the brain, it can be called a fissure. So here's the lateral fissure, which is just sort of a giant sulcus. Yeah. Okay, so that's the cerebral hemispheres. Here, just peeking out from underneath the cerebral hemispheres at the back of the brain, right, at the caudal of the brain, is where the spinal cord enters, coming in here. And then just rostral to the spinal cord is the brainstem. Yes?
On one of your previous slides, you had mentioned that a lateral section is, or a medial section is close to the midline. How close is considered close?
We'll look at that in a moment. I'll come to that. That's right, because we'll see some examples of sections. Okay. Get back to that. Okay, and then peeking out here from underneath the cerebral hemisphere is something called the cerebellum. And the cerebellum, as you can see, is another sort of highly wrinkled structure. It has a very large surface area. And the cerebellum is involved in the control of movement, particularly of fine movements. And we humans have a particularly well-developed cerebellum. Okay, so now we're going to look at a medial sagittal section of the brain. All right, what do I mean by that? Well, it's a sagittal section, which means it's going to be coming through here, and is going to show us a sort of a flat-on view of, say, the left side of the brain, or the right side of the brain. Okay. And also, it's a medial sagittal section, which means that the two cuts remain close to the midline of the brain here. So again, this would be as if you took a chopper and cut your brain right through the eyes, right to the back, and then divided it into two halves. Alright, be the sagittal section around the midline. A lateral sagittal section would be a section that was taken out here to the side. A section that didn't go through the center of the brain, that started over here, for instance. Okay. So here's a medial sagittal section of the brain. So this is the cerebellum now, and you can see how beautifully folded it is, of this very large surface area. This is one of the cerebral hemispheres. And you're looking at the inner surface here of the cerebral hemisphere, because you've chopped the brain in two, looking at the inside of the cerebral hemisphere. And let's go through some structures here. Let's start here. Here's the cerebellum. Here's the spinal cord. Just north of the spinal cord are three structures that comprise the brainstem: the midbrain, the pons, and the medulla here. And these are involved in respiration, control of respiration, and in the control of movement: the pons, the medulla, and the midbrain. And there are also, some of these areas are sensory relay stations as well. Then further north here, as it were, further in the superior direction up here, you have the diencephalon. And the diencephalon consists of the thalamus and the hypothalamus. And they're not easy to see in this section. We'll look at some other sections later on. But that's the diencephalon. The diencephalon in this area is often referred to as the midbrain. This upper area here is referred to as the midbrain. And then overlying the diencephalon is this sort of white, what's shown as a white area here. And these are all the nerve fibers that are carrying information between the two cerebral hemispheres. And this is called the corpus callosum. So it's a bundle of nerve fibers that is carrying information from one cerebral hemisphere over to the other cerebral hemisphere. And it's white because a lot of the axons are myelinated. And so the myelin is a fatty substance. It's basically lipid bilayer rolled around the axon. And so it shows up as a sort of white fatty material. And it will also show up very differently on an MRI scan because there's much less water where there's more fat. The anterior commissure is another bundle of fibers that is linking the two hemispheres. And then above the corpus callosum is the cerebrum itself here. And again, with its sulcuses and its gyruses. So that's the midbrain, the function, the sagittal section. The thalamus in the diencephalon is involved in sensory information processing to a large extent, and the hypothalamus is integrally involved in hormonal control of hormones by the brain. So those are a few basic functions. Now we're going to look in more detail at the cortex, or the cerebrum. And the outer layer of the cerebrum here, the cerebral hemispheres, is called the cortex. Right? So often the word cerebrum or cerebral hemispheres and cortex are used interchangeably. Right? But strictly the cortex refers to the rind of the outer rind here of the cerebral hemisphere, where the neuronal cell bodies are. It doesn't strictly refer to the underlying neurons in the white matter down here. So when we talk about cortical lobes, we're talking about the arrangements of the structure, the lobe-like structure, the divisions of the cerebral hemispheres. And there are 4 cortical lobes. There's an occipital lobe, and the occipital lobe is at the back here of the brain, and the caudal end of the brain. There is a parietal lobe here, which is in the middle, and sort of somewhat superior here, the parietal lobe. There is a frontal lobe up here, and then the temporal lobe is underneath the frontal and the parietal lobes. So this is what we would see if you were looking at on the surface of the brain with the cerebellum here, and this is what you'd see if you took a section through the brain, as we've done before, a sagittal section, which would show you the sort of inside of these structures. So each of these areas of the brain is specialized in processing different kinds of information. The occipital lobe back here is involved in processing vision. Kind of odd, since your eyes are out here in the front, but that's the way it is, right? So if you get a blow to the back of the head, you may see stars for a good reason. All right? The parietal lobe up here is involved in processing touch and pain sensation. Those are collectively called somatosensation. The frontal lobe is rather mysterious in many respects, but it's certainly involved in motor planning. And the temporal lobe is certainly involved in processing hearing and in language, and also in some aspects of vision as well. So the information from the ears certainly is processed in the temporal lobe, but other sensory information as well. So let's look at some coronal sections, and we'll be seeing some of the structures we've seen before, plus a few others. So again, what's happening here is a coronal section, remember, goes like this, right, or like that. It's like a bagel slicer. And so the coronal section in this slide, we're going to see 2 coronal sections. One is a section through E here on one side of the midline, and the other section is going through F here on this side of the midline. And when you look at them, you see a cross-section of the brain now with the right and the left hemispheres on either side. Okay. So now you can very beautifully see the gyri and the sulci here. You can see the cerebral cortex, this rind of grey matter of the neurons, neuronal cell bodies, and the white matter of the axons underneath. Right, so white matter is axons, grey matter is the neuronal cell bodies. And so you can see that the cerebral cortex is covered on its outer surface with neuronal cell bodies. And it's not surprising, therefore, that this is where much of the information processing in our brains occurs. You can see the fiber tracks now that run, the anterior commissure, for instance, that run from the diencephalon out to innervate both cerebral cortices. So these are the axons. These are some of the axons that are coming in to make connections to the cerebral cortex from the diencephalon in the midbrain. And in the center here, on either side of the midline, you can see some very prominent blocks of grey matter, of cell bodies, and these are collectively known as the basal ganglia. The basal ganglia. That will be on the slide here, the basal ganglia here. And they're involved in motor planning, and you've got the caudate, the putamen, and the globus pallidus as well, which I can't see on this diagram. Maybe we'll see it on another one. Okay, so these are areas that are involved in motor planning. And in some respects, they're sort of twins in their function to the cerebellum. Damage to either will produce gross deficiencies in motor control. Right? This is where Parkinson's disease has its origin, in defects in the basal ganglia. Also down here you can see the amygdala. This is actually part of the cerebral cortex, but a fairly sort of evolutionarily ancient part down here. And the amygdala is involved in emotion, in controlling emotion, right? And in controlling memory to some extent. And then the optic chiasm is where the nerves from the eyes mix and mingle and project out to the two cerebral hemispheres at the back, to the occipital lobe. We'll talk about that in much more detail later on. So we've talked about these. Let's look at now the hippocampus and the thalamus, and I think you see them in the other section here of the brain, a little further back. Here's the hippocampus, which sits just behind the amygdala. And the hippocampus is a structure that is intimately involved in memory formation and in memory retention. This is the area that was damaged in the famous patient who's known by their initials HM. And HM was able to remember everything before a certain date. So they knew who they were and where they lived. But they had no ability to lay down memory after that date, except on a very short-term basis. So you could come and visit with them and describe yourself and what you did and who you were one day, and you'd have a conversation, the content of which was kind of rooted in the past. You could tell them things about the present. And the next day, they wouldn't remember you. And they wouldn't remember what you talked about either. So a whole new day every day for HM, who had a hippocampal lesions here. Okay, so memory. Are there any other parts here? There's the globus pallidus, another little part of grey matter in the basal ganglia. And this is movement. And here's the thalamus. This last block of grey matter here is the sensory information processing, or sensory information processing. Somatosensation and vision all go through this sort of clearinghouse for sensory information. And then you can also see here the ventricles. There are these fluid-filled sacs inside the brain, and the ventricles, amongst other things, are filled with cerebrospinal fluid, and amongst other things, they provide an incompressible cushion in the center of the brain that helps the brain to resist structural blows. Okay, so that's the easy part of the lecture. Alright, there's all those structures, because I know that you love structures, and I know that the vast majority of you can remember them like crazy, much better than I can. So I'm sure that in a week or so, you'll be able to reel off dorsal and ventral and rostral and caudal much better than I ever can. Get it right every time, alright? And if you don't retain the information about these various structures and where they are till you learn them for the first exam and then you just forget about them, don't worry, we'll be revisiting them. And by the time we finish the course, you should have have a sort of pretty good intuitive grasp of what all these things do and where they are. So just grit your teeth and learn them like by rote for now, and then we'll come back and deal with them properly. Okay, so the rest of the lecture, the next 35 minutes or so, concerns the vexed subject of electricity, all right? And the thing is that one of the main things we can do in the course is to try and sort of work out the relationship between something as simple as this battery here, or a wire resistor, or a wire capacitor, an electrical component. Something as simple as this, and the functioning of our brain. And you might say, well, what's the point of that? But it turns out you can actually get quite a long way by applying the principles that you can derive from studying this guy here, right? And there aren't that many principles to apply. Most of the one's understanding of the brain at this level and a bit beyond can be accomplished with knowledge of a handful of things about electricity and electrical circuits. It's really just a handful of bits of information, all right? But you do have to have those bits fairly solid. Otherwise, a lot of neuroscience just seems rather arbitrary. So, I'm going to go through that. Now, I know that, we've got a mixed class here. Some people have had physics, the physics class. I'm not going to ask for show of hands. Some people are taking the physics class right now. And through some means, maybe AP exams, or just that you didn't realize there was a pre or co-requisite or whatever, some of you didn't take either, all right? And so you're either reviewing physics for the first time, or this may actually be the first time we talked about just basic electrical concepts, all right? So we're going to try and, just review this material, and some of you might be bored, and some of you might be frightened, and some of you somewhere in the middle, all right? And then after we've gone through it, probably after the first exam or so, when we start actually we've learned a bit about neurons. I thought I'd gather a group together of people who've taken the physics class here, people who are doing it as a co-rec, and people who didn't. And just gather together a little group, and we can talk about what works and what didn't in terms of the application of the physics here, right? Because this is the big change about putting this at the 300 level, is that before we just insisted that everybody had physics, and was sort of done with it. All right? Now we can't do that. Okay, so let's start. So I actually wrote for the 400 level class, and I think it's just as good for you. I wrote a little pamphlet called "Bluff Your Way in Electronics," right, which lays out some very simple principles. And for the 400 level class, I'd simply hand it out and say, "This is your review. Get on with it." But for this class, we're actually going to spend at least half a lecture going through these concepts on the board, right? But this is background material because your textbook evades the subject of electrical theory, right? It just does. And that's rather shocking considering that's how the brain works. The textbook is built to sell, and it's built to sell to medical students, and medical students are notoriously adverse to physics. Yeah. All right, so I'm not a medic. I'm a scientist, a researcher, and so I'm not averse to physics, although I'm not very good at it. All right, so I will say things that just seem dumb. If I do, stick your hand up and say, that's not the way I learned it, and we can have a discussion. All right, on a discussion board. I don't care. I've learned enough to survive in neuroscience. I'm happy to pass the information on. It may not be 100% pure physics. All right? But I'll try. Okay, so let's start. So electrical charge, right? So the essential thing about electricity, I think, is electrical charge. Because electrical charge is a fundamental property of many kinds of subatomic particles. It's a fundamental property of electrons. It's a fundamental property of protons, for instance. All right? So electrons are negatively charged, and sodium chloride, for instance, is composed of negatively charged chloride ions and positively charged sodium ions. All right? So charge is basic property. And I don't know whether we'll use this or not later in the course, but charge is measured in units called coulombs, for what it's worth, after Monsieur Coulomb, I think. Am I right? Anybody know? What's Monsieur Coulomb? Yeah? Yeah, right. Monsieur Coulomb. And a single sodium or chloride ion has a charge of plus or minus 1.6 times 10 to the minus 19 coulombs. So a coulomb is a really big unit, right, compared with an atomic charge. And we won't ever be dealing in coulombs. So, if you look at a solution, right, like let's take a bucket of sodium chloride. Okay, here's a bucket of sodium chloride, right? Okay. If we look at a bucket of sodium chloride, there are as many positive as there are negative ions in that solution. Right? The bucket is not charged. Right? The number of positive ions cancel out the number of negative ions. There is no net charge. So the overall charge at any one sort of area, maybe not point, maybe that's wrong, maybe an area within the solution is zero, the net charge. Right? Yes? Yeah? Alright, good. I need some encouragement here. Otherwise I'll fold up and start crying. And this is called the principle of electroneutrality as it relates to solutions. It just means that in a small part of a solution, generally, there's no net charge. The number of bulk chloride ions in a sodium chloride solution is equal to the number of bulk sodium ions. Alright? Now, if you go down to a teeny tiny microscopic level here, an atomic level, that's not going to be true. But by and large, for all intents and purposes, it is. Now, separating these charges, right, getting all the sodium ions over here, and all the chloride ions over here, separating out those charges over any appreciable different distance. What do I mean by appreciable distance? I mean even a matter of angstrom units. Right? Separating out charge in a bulk solution takes an enormous amount of energy. It takes a lot of energy to separate the sodium and chloride ions in this solution. So, positive and negative ions can only be separated in solution in a very local, very small distances. Right, you can separate these out, but only in a very sort of local area, like that. Right? Can't take all the sodium ions and throw them on one side of the bucket, and all the chloride ions. It takes way too much energy to do that. And even so, with the energy that's normally available in thermal energy or in biological systems, only a tiny percentage of the ions can be separated like this. Only a tiny percentage of the ions can be separated. So, electroneutrality really is not violated, on the whole, by this minute separation of a very tiny percentage of the available ions. Right? On the whole, in the bulk solution, there's still as many positive as there are negative ions. Right? So, in the bulk solution, there's still as many positive as negative ions. But in this little microscopic area, we've managed to separate. So, just such a separation of a tiny fraction of the ions across a very small distance occurs across cell membranes. So the cytoplasm inside a cell can carry more or less charge than the neighboring extracellular space. So you have a cell now, and it is possible that negative ions just at the surface of the membrane here, there is a preponderance of a few more positive ions, for instance, or negative ions on one side of the membrane than the other. But then that's surrounded by a sea of ions outside and a sea of ions inside, where the positive and the negative ions are equal and opposite in numbers. Alright? Okay, so when you look in a textbook and you see sort of pictures of sodium ions rushing in through channels, it gives you the impression that all these sodium ions go into the cell, and what's inside the cell are a bunch of sodium ions, and what's left outside the cell are a bunch of chloride ions or anions. Right? That's not true at all. What goes into the cell is a tiny fraction of the sodium ions that are outside the cell or inside the cell. Right? By and large, there are as many sodium and chloride ions on the whole, but there's a little tiny imbalance. And that imbalance, tiny as it is, creates a charge difference across the membrane. So that one side of the membrane is positively charged with respect to the other. Okay, so that's the first lesson. So what is this separation of charge that can be achieved across cell membranes? And we'll talk about how it's achieved later on. But you just have to take it for granted now that it can be achieved. And in 207 and 330, you've covered how it's achieved, right? Through pumps and channels. This separation of charge creates an electrical potential difference across the membrane. So when charges are separated, an electrical potential is said to exist between the 2 points in space to which the charges have been moved. So there is an electrical potential difference between this side of the membrane, in this case, and that side of the membrane that's created by this tiny disparity of charge. Since separating the charges, as we've pointed out, requires energy, requires work, electrical potential is stored energy. So electrical potential is a measure of energy. And you can think of it as being stored in this gradient here of charge. So that's why the separation of charge requires, for instance, ATP hydrolysis and pumps, for instance, in some cells, in bacterial cells. And so this now is called the membrane potential, the potential across the cell membrane. Electrical potential is measured in volts, so we had coulombs, so now volts, and it's called voltage. So the potential difference in volts between 2 points is an energy term. It's the energy needed in joules to move a charge of 1 coulomb between the points. Stupid definition. We'll never come across it again. I promise. But it's important you realize that electrical potential is energy, stored energy. And membrane potentials are measured, this is important, in millivolts. Right, so this battery here is a 9-volt battery. The potential across a cell membrane is about 100th of that. Batteries are devices that can use chemical reactions, so little chemical reactions can occur in here, to separate charges in the solution that's inside, or whatever it is that's inside. And this creates, on the battery, it creates a positive terminal and a negative terminal. It creates a potential difference between the two terminals, and that is a source of energy. So that if I connect a wire between those two terminals, I can release that energy. I can make that energy do work. Right. So that's what a battery is. Stored energy. Okay, so we've got a positive terminal here where positive charge can accumulate, and a negative charge terminal over here. And if we put a wire between them, there will be a current flow between them, because like charges, the wire contains electrons. The electrons are charged, and so the electrons will move towards the plus terminal and away from the minus terminal. Right? Like charges repel, unlike charges attract. And so if I put a wire has a metal and all these electrons associated with the metal atoms, some of those electrons will start to drift towards one of the terminals, and that creates a current. So positively charged particles tend to move away from areas of positive potential towards areas with negative potential. And this flow of charge constitutes an electrical current. And it is a convention that electrical current flows from the plus to the minus. That is a convention. Right? We declare it to be so. Right? The electrons are actually drifting in the opposite direction. But we declare that positive current flow is from plus to minus. Right? Okay, so the battery, positive current flows from plus to minus. If the current was able to move in the other direction, it would be called negative current flow. So batteries can drive current around wires connected to their terminals. And current is measured in amps. And the amp is a measure of the amount of charge that moves through the wire per second. Right? How many coulombs move through the wire per second. And again, in cells, tiny currents flow. 10 to the minus 9 amps is a typical nanoamp, is a typical current that would flow through a cell membrane when the neuron is active. 10 to the minus 12 amps, a picoamp of current, might flow through a single ion channel. Okay, so neurons generate, the membrane potential here can drive tiny amounts of current across the cell membrane and around the cells. Now, one of the purest things about current, I'm not going to read all this, one of the purest things about current is that the electrons, or in a solution, it's the ions that carry the current, as it were, move a lot slower than the energy of the current itself. And what that means physically is, if you're in a horror film and you're taking a bath, the actress is taking a bath, horror film, and the villain comes in and throws a toaster into the bath. All right, they get electrocuted immediately, right? Nobody's going to wait for the ions to diffuse from the toaster to the person, because it would take hours for individual sodium ions. Even if they were in a bath of sodium chloride, it would still take hours for individual sodium ion to diffuse from the toaster to the other end of the bath. Yeah? And so, but when the toaster hits the bath, current flows, right, from the toaster into the bath, and immediately current flows at the other end and flows through the heroin and toaster stove. All right? When you come into a room, you switch on a light. Right? You switch the light, immediately the light bulb goes on in the other end of the room. But the electrons have hardly moved physically, have hardly drifted at all down the wire. Okay? One way of thinking of it, so the energy, electrical energy, and the current movement, as it were, the movement of the energy, moves at the speed of light. Remember, the speed of light is different depending on which medium it's happening in. But it moves roughly what we call the speed of light. Instantaneous. So if current begins to enter the bath at one end, or the wire at one end, it instantaneously emerges, starts to flow at the other end. Instantaneously. One way of looking at this is if you think of billiard balls. You line up a whole bunch of billiard balls so they touch. And you take a billiard cue and you hit one end of the row of billiard balls. And the billiard ball at the far end immediately pops out. Alright? And that's because a wave of energy has passed along the billiard balls. But the billiard balls themselves have hardly moved. Each single billiard ball hardly moved. The same with sound waves, right? You don't think when I speak here, the sound wave goes, sound energy moves as a wave at, 700 miles per hour or something down the room. You don't think the molecules that emerge from my mouth move that fast. I hope not. Okay, so that's what's happening here. So electrical energy is transferred as an electromagnetic wave of light speed. And this is important for your understanding of what happens inside cells. So sodium ions, for instance, enter a cell through a channel, an ion channel, right? And they carry through that ion channel a current, an electrical current. And then that electrical current will spread along the cell. But it isn't those sodium ions that entered that's carrying, that's diffusing down the cell. It's the electrical current that's moving down the cell. It's the energy of the electrical current that's moving down the cell. So don't think of an action potential, for instance, as a diffusion of sodium ions carrying positive charge from one channel to the next. That isn't what's happening. The sodium ions are carrying electrical current, and the current is much more rapidly moving along the axon. And what could be carrying the charge within the cell could be chloride ions, it could be electrons, it could be all sorts of stuff. Right? Not those sodium ions that came in. So when you think about nerves, don't count the ions. Don't try and think of the ions diffusing. Think of the currents that flow. And realize the reasons the current is flowing is because the ions are drifting into the cell, or out of the cell, or through the solution. Okay. In the same way as you don't think of what the individual electrons are doing when you think about wires. Electronics. Okay, so one of the things about electrical currents and charge is that it can't be created de novo, it can't be destroyed. So once a current begins to flow, it has to continue flowing around the circuit. Alright, so current tends to flow in circuits. That means that if current leaves a cell, for instance, current leaves a cell through a channel, then it's got to flow back into the cell somewhere else. Right? Current flows in circuits. It doesn't just vanish. Alright. So the next thing is to try and tackle these concepts of currents and voltage in relation to natural objects. So the two basic sort of components, electrical components, that we use to model, three actually, to model the behavior of cells are resistors and capacitors and batteries. Those are the only three. And that's the only electronics. You're going to learn about transistors, you don't have to learn anything else. Just resistors, capacitors, and batteries. So let's start with a resistor. And a resistor has an electrical symbol that looks like this. And if we apply a potential difference across the resistor, current flows through it. Alright? And there's something called Ohm's law that tells us how much current flows through if we apply a certain amount of voltage across this resistor. And Ohm's law is really simple. It just says that the amount of current that flows is inversely proportional to the magnitude of the resistor. So if the resistor is twice as long, twice as big, if it has twice as much wire in it, if it resists twice as much, then half as much current flows. That's Ohm's law. That's why it's called a resistor, right? Because it resists current flow. Okay? So if we know the value of a potential difference across a resistor, we can calculate the current that's flowing through it using Ohm's law. And we can also do the converse, right? If we measure that a certain amount of current is flowing through a resistor, we can use Ohm's law to calculate what the potential difference across the resistor, and it's called the potential drop across the resistor is. Right? That's just the converse, right? And so that's using Ohm's law in this way, that potential drop across a resistor is equal to current times the resistance. So the higher the resistance, the more potential drop. And it's actually this form, perhaps, which is the most important for neuroscience. Because in neuroscience, what we're often doing is saying a certain amount of current is coming through an ion channel. What is its effect going to be on the membrane potential of the cell? Right, that's a question that we often ask in neuroscience. What is the effect of current flowing through an ion channel going to be on membrane potential? Okay. Now, resistors do not allow charge to accumulate along their length. So the current flows in, and the current flows, the same amount of current flows out. Right? No charge accumulates in a resistor. Important. Okay. And examples of resistors are metal wires, where the charge is carried by electrons, or ionic solutions, like this sodium chloride solution here, where the charge is carried by sodium and chloride ions. The current. Okay. Now, capacitors, which are represented by this kind of symbol, are, in some respects, sort of, in a way, they're sort of an opposite to resistors, because I mentioned that resistors don't allow charge to accumulate, but they allow current to flow. Capacitors store charge. So capacitors are capable of storing charge. That's why they're shown as if they were two plates, two metal plates, separated by an insulating material. Because one side can store a lot of positive charge. And when they're fully charged, they don't allow any current to flow through them. Once the capacitor is so-called fully charged, it won't allow current to flow through. And so in that sense, they're very different from resistors. So if a capacitor is connected to a battery, here's our battery. This is a symbol for a battery here. This long bar is the plus terminal, and this is the negative terminal of the battery. So if we connect a capacitor to a battery, we close the switch here, then the capacitor becomes charged. This little line actually shouldn't be there. I'll get rid of that. Notes. So you close the switch and the capacitor becomes charged. And when it's fully charged, no more current flows through the circuit. So current will flow through the circuit, and the current will deposit positive charge from plus to minus. The current will flow in this direction. And as it flows, it will deposit positive charge on this plate of the capacitor. And then when the capacitor is fully charged, when its voltage here equals the voltage of the battery, then no more current flows. That's it. So capacitors are devices that store charge. Now, I'm not going to go into these two examples here of calculations, because they're just calculations and you can do them. We can run through them at the beginning of the next time. But what I want to finish the lecture with is just with cell membranes. What's the relationship between capacitors and resistors and cell membranes? So firstly, the extracellular space, right? Or the intracellular space, the cytoplasm of a cell. The cytoplasm of the cell, or the extracellular space, functions as sort of a three-dimensional resistor, right? It allows current to flow, but it resists current flow to some small extent. So the extracellular fluid, or the intracellular cytoplasm, can be represented electrically by a small resistor. As current flows through it, a little bit of potential change will build up. And the amount of resistance depends upon the geometry. Thin dendrites will have high resistance. Thin axons will have high resistance inside, because it's more difficult to force current through a thin dendrite than a thick dendrite, or a thin axon versus a thick axon. That's like water. You need more pressure, you need more voltage to force current down. The lipid bilayer of the cell membrane functions as a capacitor. And it's easy to see why. If you think of the inside here of the capacitor as the lipid insulating hydrophobic core, and the two sides of the capacitor as the plates of the capacitor as the phospholipid head groups, then it's easy to see why the lipid bilayer functions like a capacitor. Charge can be stored on the surface of the lipid bilayer. What about ion channels? Ion channels can obviously be represented by a resistor. Right? The resistor represents as the current flows through the pore, it encounters a resistance. So one part of an ion channel is going to be represented by a resistor. But there's another aspect to the flux of ions through a channel, and that is the driving force, the electrochemical gradient that's driving ions through the channel. The reason why sodium goes into cells and potassium comes out of cells. And that electrochemical driving force is represented by a battery. So this is a membrane. It has a bilayer. It has sodium channels. It has potassium channels. And this is the electrical model of that cell membrane. It has a capacitance that represents the bilayer. It has a resistance here that represents the pore of the potassium channel, and a resistance here that represents the pore of the sodium channel. And it has two batteries here that represent the force that's driving ions through that channel. In the case of sodium, that force, as you know, drives sodium ions into cells. In the case of potassium, because it drives potassium out of cells. Because there's more sodium outside cells than inside, and more potassium inside cells than out. And so that's the reason why these batteries are inverted. Right, because of the different driving force for ion movements through the channels. Okay. So, believe me, if you can think of neurons as being little electrical circuits or neuronal membranes as little electrical circuits, and think not just in terms of which ion goes where, the sodium ions rush in, the potassium ions rush out. If you can deconstruct these membranes into tiny electrical circuits like this, you can understand why neurons function the way they do at a much deeper level. So that's what we're going to try and do. Okay, so read this material here. Ask me questions on the discussion page. Ask me questions the next meeting. As you can see, the anatomy is straightforward, but this stuff is not.